Hi, I'm Lynn Chang, and I'm Professor of Medicine at the David Geffen School of Medicine at UCLA. I'm also Vice Chief of the Vache and Tamar Manoukian Division of Digestive Diseases and Co-Director of the G. Oppenheimer Center for Neurobiology of Stress and Resilience.
Hi, I'm Beth Videlock, and I'm a clinical instructor of medicine at UCLA and the lead author, and Dr. Chang is the senior author, on the paper published in the American Journal of Physiology entitled Sigmoid colon mucosal gene expression supports alterations of neuronal signaling in irritable bowel syndrome with constipation. We hope that you will take a look after seeing this brief introduction.
Irritable bowel syndrome, or IBS, is a prevalent functional gastrointestinal disorder characterized by abdominal pain associated with alterations in stool form or frequency. We perform gene expression profiling analysis by microarray on sigmoid mucosal biopsies collected from 20 individuals with IBS and 10 healthy controls, which we'll refer to as HCs.
This sample was carefully designed, with equal numbers of men and women in each group, and with limited medication use and no use of SSRIs or neuromodulators. We can feel more confident that any group differences we see in gene expression are not due to sex or IBS treatment. In addition to looking at transcripts that were differentially expressed between IBS and controls, we performed weighted gene co-expression network analysis, or WGCNA. WGCNA uses unsupervised clustering to identify highly correlated genes. Now we can look at the whole microarray summarized by modules to see if any are related to IBS status or symptoms. Module names are overrepresented gene ontology terms, or KEGG pathways. In the paper, we take a deeper look at the cyclic AMP protein kinase A module, which is associated with symptoms in addition to IBS status. This module, in combination with other findings described in the paper, support our conclusion that neuronal signaling is altered in IBS.
Beth, I think it's important for you to describe the effort you took to present robust and reproducible results.
Sure. I've always been struck by how often divergent results are published in IBS.
It's true, but there can be multiple factors that can contribute to conflicting results.
Well, I decided to take advantage of the public data repositories to see if I could compare our results to results from other microarray studies. I found 2 studies with microarray data, one from the Mayo Clinic and one from the University of Nottingham. Among the transcripts that we found to be differentially expressed between IBS with constipation and controls, 859 were detected by probes in the microarray platform used by the Mayo Clinic and Nottingham. Of these, the number differentially expressed in the same direction as in our study were 57 for Mayo, 185 for Nottingham, and 17 for all 3. I focused on Mayo because this study, like ours, looked at sigmoid colon. Commonly dysregulated genes included genes involved in stress and neuropeptide signaling, immune and barrier function, and neurite outgrowth. I wanted to facilitate review of this gene expression data for researchers in the field, so I created a website that will display results from our study, Mayo, and Nottingham for any gene present on the arrays. Go to uclacns.org/IBS microarray comparison and enter the Entree gene ID or symbol for any gene. You will see a table with data for each comparison: IBS, IBS with diarrhea, and IBS with constipation versus control in each cohort. Below the table, there are dot plots showing the data for each probe in all 3 cohorts.
We hope that you find this resource useful. Please read the paper to learn more about our gene expression profiling study in IBS, in which group differences were found predominantly in IBS with constipation versus controls, and which supports alterations in neuronal signaling in IBS.